sites. But you never chase the blue lines, right? But I would say some clear TV works right outside of.
Yeah, plus,
You never need potatoes.
the people who implemented it are skilled at rapid prototyping and not at diligence. And attention to detail. Right? So I'm not even sure it was implemented right. There's Matt and I are talking about incorporating into the bake off, right? So with the color aggressive stuff.
That's actually on the ethos paper, which he said works. So for zero shot generation, he says they're getting good results from it, even really long term predictions, which is really interesting. So that basically removes fine tuning as a requirement from a model.
And who's the main group behind ethos?
If you're interested in Asia too, I can recommend.
I think we'll have with at Columbia, they've almost got permission to release their model weights, which is
Nice.
competition. So sometimes got shut down by them. So we'll see whatever gets picked back up. Jar. Influence the health policy. It was the insurance, they actually had the insurance policies that let's start at the. Oh, that's right. It's like bottom. Oh, sorry.
Have you talked to them?
You went to
about,
5 years.
maybe we do academics somewhere else? Because you want to be an American.
There's also a spot there.
Yeah, even though. It's the best place to do research. Yes.
Yeah.
I mean, what's the one on the air? It's not anywhere else.
Yeah, there's a lot of jobs.
Harvard.
Some use volume jobs, but it's a lot of tough to use, like.
And I did the interview.
Yeah.
So.
It's just an EHR model with Temple.
Health services.
Basically what Bay Area has.
The speak to 2 hour linearization. And then instead of building a classifier, you have your label function. You sample generations to some time horizon. So you generate up until the next date. So you generate like 100 examples. Then you would train a lot of community to find your labeling function and you estimate the probability of your outcome based on the label. So as soon as you have a good labeling model and it's trivial, so it makes a bunch of hand-wavy stuff. And it seemed like it would not work. You're waiting. And then good to see you. Morno implemented some tasks. In charge, surprisingly. I'm just gonna watch. It's interesting. It's an interesting way to think about using it. Never. It's always very fine too. And it's just basically based on the label definition, which makes it like amenable to basically generate data by our labeling function. And that's just count how many in the soup with that's what they do.
That's why I'm saying.
They said that was all the papers except ours are really hard to run the trade. They're not very lazy. What's cool about this is it plugs you into scaling. How far down can they predict it? Well, that's where I'm rating. Like, you read the paper, when I read it, like,
So actually get out of calibration really fast. Yeah, that's when like the UV amenables are actually faking it on.
Everything else but this.
Yeah, okay, so let's begin. I'm not as loud as Jonathan, but I'll try to. So, hello everyone, let's begin. Welcome to the Beamer Colloquium. Jonathan looks different today. So, you know, we have with us today Elior Rahmani, who's an assistant adjunct professor at UCLA. And yeah, I'll try to do Jonathan's script as we do. So before we begin, we ask everyone about, how many tickets you have. So, where did you grow up? How did you end up being here? What was your career path like? So if you could share for a couple of minutes, more on that, and then maybe we can talk. So
yeah.
Over to you.
Okay. I think it was maybe one speeding ticket. And, I was born and grew up in Israel. I started my bachelor's degree at Tel Aviv University, continued to UCLA, got my Ph.D. in computer science, then moved to UC Berkeley for a postdoc. A couple of years ago, got back to UCLA as part of their computational medicine department. And yeah, I guess the science part, that's the fun part, right? Ready when you are. Yeah.
Go ahead, yeah.
All right. Okay, so as I just said, my background is in computer science, and very naturally I take a computational approach to biomedicine. And more specifically, my focus is really about developing statistical machine learning methods for analyzing genomic and clinical data. And more specifically, I can beam my work roughly into those three beams. And this is a representative list of my work. And the first beam to the left is how can we take those high-dimensional data sets and identify those biases, those hidden biases that we have in the data and account for them, essentially accounting for unknown and hidden confounders. The second beam is about how can we take those large clinical genomic data sets that collect bulk level, tissue level genomics and infer something at the cell type level from the bulk. And the third beam is about how can we identify disease subtypes or patient subgroups based on, say, genomic or clinical data. And over the years, as I was developing this method, I became more and more interested in working with clinicians, making sure that I actually work on questions, clinical questions that can eventually matter and translate into clinical practice. And when we think about those types of questions, oftentimes we can beam them roughly into those three types of questions. First type is about the identification or classification of disease or diagnosis. Here's one example for that. We can take whole blood DNA methylation, try to classify rheumatoid arthritis patients from healthy individuals based on the methylation. Another type of question we can ask are about the course of disease, prognosis. And here's an example of taking data from all of us, trying to predict whether glaucoma patients, whether a glaucoma patient is going to end up needing a surgery for the glaucoma condition. The third type of question is about therapeutics. For example, can we predict drug response? One example here, you can take lung function metrics and try to predict whether an asthma patient is going to respond to drug. Now, every time we develop those methods and those models, we really think about potentially deploying it and affecting patients' lives. And when we think about that, what we really care about is first, we want the models to be accurate. But second, we really need the models to be generalizable. And this is actually very, very challenging in biomedical data analysis. And what exactly do I mean by generalizable? First, we want the models to be replicable, meaning we want them to be robust across independent data sets. And in the example to the left is rheumatoid arthritis. This is actually taking whole body inoculation from European samples, taking all of the, using all the best practices we know, cross-validation, right, and so on. And what you see here is just the performance on the held out data. But once we take another cohort, also European samples, also whole blood, and apply the model, we see that the performance is actually not better than random. Another thing we want from our models is to be portable, meaning we want them to be robust across location. In the example in the middle here, this performance that you see here is actually based on one part of the United States. what happens when we try to transfer the model to another part of the United States? Another drastic decrease in the AUC. And we also want our models to be equitable, meaning we actually want them to be robust across populations. And the model here of the asthma drug response to the right was actually learned based on Mexican samples. And once we apply it to African-American samples, again, a significant drop in AUC. Okay, so clearly there is a problem here of biases in the data. A simple, random test train split is just not good enough, and you need to tackle it. But when you think about it, the big challenge here is not only how we evaluate the models in a rigorous way to make sure they generalize, but actually how do we learn those models? How do we train them to be generalizable and accurate at the same time? And I think that is really the fundamental questions in biomedical data analysis. Now, we might think at that point, maybe, AI can come to our rescue. There have been quite a few, as everyone knows, successful applications. But one big difference is if you go back to Stats 101, the way most of the applications in biomedicine still work is we have the entire population of patients of interest, and for practical logistical reasons, we only subsample a subset of them. That's our data, and then we infer a model based on that. Now, I've shown you a few examples, and there are many other examples where models just don't generalize. What essentially it means is that those models that don't generalize are fed to the data and specifically biases in the data and don't necessarily generalize to the entire population. Now, when you think about a successful model, AI model such as ChatGPT, right? Essentially what it does, it learns a model for the entire written English language. Now, when they actually crawl the web and take essentially all the written English language, what they do is actually feed the model directly to the entire population, not a subset of the data. So this is a very important distinction to make between those successful AI applications and many of the biomedical analysis applications. Now, my conclusion based on that is I really want to explicitly address the sources of inconsistencies in those biomedical applications. And there can be many such sources, many of which are actually things we can control with just being more careful, maybe more resources. But there are a few that I'm going to focus on that are more inherent to the way we collect the data and analyze the data. First one is about unknown confounders. So maybe we collect our controls and cases in a study, and here represented by the gray and red colors. Now we know that different individuals are not just different by many different factors, genetic, environmental, clinical conditions, and so on. Some of those variables, even though we try to collect them the best we can when we design studies, some of those variables will always be unknown. And when the distribution of these variables are different between the case and controls, that's where we're risking at risk of capturing those confounders and models not being consistent. Another thing that can lead to a lack of consistency between studies is the fact that complex diseases are oftentimes known or at least suspected to be very heterogeneous. Maybe they are disease subtypes. Now imagine that those cases are actually not just the same type of case. Now, say we have these two cohorts corresponding to two disease subtypes. That distribution of how many we have of subtype 1 versus subtype 2 can really differ between different studies studying the same condition. So that's another source of inconsistency between studies. And the third one is something I call hidden dimension. And when you think about it, most of our data sets are still in this tabular form. We have these samples by feature data. But oftentimes in biology, we actually have those hidden dimensions of information that we don't necessarily get to observe. And bulk genomics, tissue level bulk genomics is one excellent example for that. We have this hidden cell type axis dimension of information that we just don't get to see. But we know that biology acts at the cell type level. And if there are changes, if there are differences between data sets across that distribution, this hidden distribution, that's another source of inconsistency. Now, okay, so as I told you, my conclusion is that what we really want to do is to explicitly address those sources of inconsistency so that our models are more likely to become generalizable. But there is another critical distinction to make compared to those other successful stories in AI. And I'm going to show you an illustration to explain that. So I took this image of a cat, asked ChatGPT what animal is in this image, and none of you are surprised to see you got the answer right? The reason why you're not surprised is because we actually understand there is sufficient information in this input to tell us this is a cat. On the other hand, if I take the exact same image, drastically decrease the resolution, and ask the exact same question, I actually don't get the answer. And you're not surprised by that either, because your brain tells you there is insufficient information to ask if this is actually a cat or a barn owl, right? When you think about it, if you take someone's genome or medical health record, and you ask a question such as, will the patient respond to treatment? That might actually fall into the same class of insufficient information. We just don't necessarily know if we have enough data, the right data, or the right data resolution to really accurately fit a model for the entire population of patients and answer those questions accurately and confidently. So my conclusion based on that is that we really want to try to target questions that are tractable, meaning that we have enough information accurately answer those questions given the data we have in hand, the size, the type, the resolution, and so on. What do I mean by tractability here? So let's take this question as an example. Will the patient respond to treatment? And I want you to imagine this tractability spectrum. Now we know that complex diseases are involved hundreds, not thousands of factors, right? And for them, maybe that question, given the data we have in hand, is not going to be tractable. But at the same time, we have a great body of literature telling us that complex diseases are heterogeneous, right? So I wanted to think about complex disease as a tangled ball of yarn. And it might actually be composed of multiple strings because it's heterogeneous diseases. Maybe we have subgroups, right? So what if we can disentangle this ball of yarn into some of its composing pieces? Maybe some of those pieces will actually be tractable, meaning maybe we can answer that question for a subset of the population or a subtype of the disease. One example for that is there is a subpopulation of breast cancer patients for which just 4 factors make them very predictable in terms of their outcome. So I'm going to go back to this initial set of 3 questions, and I'm going to revisit it by saying there is actually this hidden layer here, right, between the disease and the question of the subtype, right, or the heterogeneity of the disease, which we don't know. But my approach is going to be, if I want accurate models, what I'm going to do is I'm going to target those subtypes for which we have enough information, enough signal in the data to answer them accurately. And I'm going to address the lack of generalizability and the reason for it by explicitly targeting those unknown subtypes, confounders, and giving them interest. And just to summarize the research goal, it's essentially about identifying tractable and clinically meaningful patient subgroups. We want to do so robustly, but importantly, we also want to do so systematically. And I already mentioned that there is a great body of literature that have shown clinically meaningful subtypes. But when we look at the methods that led to those findings, those are actually based on pretty simple methods, simple clustering methods that cannot just be applied to any data setting condition. They just typically don't work for other conditions, other situations. So we want methodology that can allow us to systematically apply this pipeline and identify those patient subgroups. Okay, what does it mean to find patient subgroups? Just breaking down to practical tasks, right? We need to find heterogeneity or variation among patients. And what does it mean to learn meaningful variation among patients? I want to start with an example from single-cell genomics. So we have the cells by gene, gene count metrics, right? We can ask questions such as, can we find cellular and molecular variation that is unique to cases? If we have the answer to that, we need to ask, can we use this variation for clinically meaningful patient stratification? Now, a typical workflow of single-cell genomics involves typically reducing the high dimension of genes into some low-dimensional latent representation, which we can then visualize, and maybe some clusters of 4. Now, that latent representation is most of the time done using unsupervised learning methods. And what those methods do best is actually capture dominant source of variation in the data. What does it mean? Essentially, what it means that to a first order approximation, it means that it captures variation that is correlated with many features in the data. So if you take, say, cancer patients, then because we know that many genes are affected by cancer, or at least correlated, associated in changes with cancer, then those clusters may correspond to meaningful variation in this cancer, right? But if you take other conditions that have more subtle variation that are intrinsic to cases, then it's more likely that those clusters will correspond to something different. And in fact, in single cell, we know that those clusters typically do correspond to cell identity, cell state, maybe even batch effects, and so on. So we're going to take a different approach. The different approach is going to be, let's use also cells from control. So healthy individuals who don't have the condition. And I'm going to use a contrastive learning approach. Now, typically when we say contrastive learning, we have a type of model in our mind where we try to learn a representation under which we try to pull apart these two groups, in that case, the case and controls. And at the same time, it will also induce low variation within each one of these clusters. But this is not what we want. What we want is actually try to find a model under which we have high variance for those red dots, which represent those cells coming from cases, and at the same time, low variation for cells coming from controls. We don't necessarily care about separation because some of the case cells, right? Some of the cells coming from those cases might actually have a healthy phenotype, right? And to kind of translate that into math, what we really want here is to find a model parameterized by theta that is going to show us high variance among cases, but low variance among controls. So we really want to find this theta that will maximize this ratio. Now, if this is our objective, what we should expect to end up getting after we visualize this representation that we learn based on this objective is this much larger spread of red points coming from cases, and at the same time, much lower variation for the points coming from controls. And what can we do with that? One thing we can do with that is, hey, maybe now if we focus on those cells coming from cases, we can actually find some cluster that is really more either specific or enriched in those cases, and now it's going to have a different interpretation. Another question we can ask, maybe if now we look at the cells from each one of the patients separately, we can identify some patterns of variation between the patients, because the spread, for example, of their cells is actually different depending on something like their condition state. And that's exactly the first example that I want to show you. We took this single cell RNA-seq data from PBMCs. We had lupus patients in this data and healthy individuals. And the first thing we did was actually to apply this idea using an existing state-of-the-art method called contrastive VI. And what you see here is basically a bunch of cells coming from either healthy or lupus patients based on they're represented by the blue and orange colors here. And what I'm going to show you now is what happens when we take all the cells, all the dots from one patient at a time, and we're summarizing each patient. So we're summarizing all the cells of every patient by just taking the variance. So we're going to take the variance along the x-axis and along the y-axis. This is the latent representation that the contrastive VI model learns. And when we do that, we get this relatively nice separation between the points representing lupus patients and points representing healthy individuals. And when you think about it, that's exactly what we wanted, right? We wanted to find representations that reveal higher variations in cases compared to control, and that's exactly what we get. So what's the problem here? The problem is that this model was trained based on European samples in the data set. Once we take the non-European samples and apply the model, it just does not replicate, right? So can we do better? So I'm showing you now how to tackle this problem from the perspective of unknown confounders. So remember, I showed you this illustration. We have the controls, we have the cases, we have those unknown confounders, those different colors that are different in distribution in this illustration between case and controls. Now, I want you to imagine, what if there is actually in this data set a subset of the controls and a subset of the cases such that they're actually balanced with respect to all the unknown confounders, right? If that was the case, and we knew these two subsets, we could just analyze the data based on these samples. We would get valid results. They will be unconfounded. It's single cell data. We don't really need a lot of samples necessarily because we have many cells per sample, right? So we may be powered enough to actually do that. Obviously, the problem, we don't know the subsets, right? But what we can do is we can anchor the results implicitly based on them by taking a randomized approach. Now, what do I mean by that? So let's say we take a random subset. So a random subset of the cases, a random subset of the controls. So this is the first subsample. Most likely it's going to be confounded in some way, right? But we can still learn a contrastive model. Maybe the ratio is going to be above 1, indicating that there is actually more variation in cases compared to controls. And maybe the spread of cells is going to look something like that. Maybe we do have more variation in cells coming from cases compared to controls. But because it's confounded, maybe there is actually some hidden confounder which we don't get to see because we don't have it in the data corresponding to these two confounder states, the green and the purple one, right? Now, what happens if we now take that model and apply it to more random subsamples of the data? So we can do that, right? Once we try to apply it to a subsample that is actually balanced with respect to those confounders, even though we don't necessarily know it's balanced, what we expect to see is that this ratio is going to be roughly 1 because it's it's balanced with respect to confounders, so it's not confounded. If the model is confounded, then there is not going to be any signal in unconfounded subsample of the data, right? Now we can do the exact same thing based on each one of the subsamples in the data. And if we learn a model based on the subsample that is actually balanced, and we find contrastive variation, then assuming the real signal does exist in all of the subsamples, maybe in addition to confounders in some of them, then when we apply it, because it's unconfounded, apply it to other subsamples, we still expect to see this contrastive variation. In other words, we can come up with this objective that we actually want this ratio to be above 1 in all of the subsamples of the data. And just to translate that into something that we can actually optimize, right? We really want the worst case, so the worst case subsample, the minimal subsample, meaning the subsample that has the minimal ratio, to be as high as possible. And that gets us to this maximum optimization problem. So the maximum is still the same as before. We maximize with respect to the parameters of the model. The minimization is with respect to those subsamples of the data. This is a type of domain generalization problem, which is it's a known class of problems, but the trick is really to understand what is the right objective and what's the minimization is with respect to. Now in practice, we don't really need to subsample explicitly those subsamples, but rather to solve this optimization problem. And to make it a smooth function, we actually change that to something that is actually a smooth function and not discrete, like what you see now. So we use a relaxed objective, but interestingly, we actually do have a theorem showing that the optimal solution for both problems is equivalent. Okay, so now let's go back to the lupus example. We still have this problem. Once we apply this RCL, the robust contrastive learning model, finally we see that we do have consistency between the European and non-Europeans. Okay, so this is nice, right? But remember, we still have concrete questions we want to ask based on that representation. Specifically, can we find clinically meaningful patient stratification? So this is lupus. One thing we can do now is to focus only on the orange points, only on the cases, and we can ask something like, why do those patients in the top right are different in their variance compared to the other patients, right? And what we found is that those patients are actually the patients that have active disease states based on the SLEDAI disease index for lupus. Another type of question we can ask is, can we find meaningful cellular and molecular variation? Maybe there is an interesting cluster, right? Now, we did do that for lupus, but I actually want to show you another example that is more challenging because lupus is a systemic level disease. I'm going to show you an example where the signals were even weaker. And specifically, I'm going to talk about naive T cells in the context of age. So now I'm going to think of aged individuals as cases, right? And young individuals as controls. We're gonna have above 60, below 40, and I'm going to try to find representation of the cells that will reveal heterogeneity that is really more enriched with age. Okay, now I'm going to use the exact same data sets. So we have both lupus cases and controls, but because the method implicitly accounts for all the confounders, I'm not gonna care about that. I'm not gonna even model that, right? All the confounders are going to be kind of automatically taken care of. And we learned the model based on CD4 naive T cells, and we learned specifically 10 components. And what you see here is the UMA based on those 10 components. And you can already see there are a bunch of clusters here, right? The first thing you do when you have those clusters is basically try to ask, okay, what gene is most differentially expressed in that cluster compared to the others? So we did that. And one thing that was really standing out at that point is we actually have a cluster of CTLA-4 expressing naive T cells. And when you ask an immunologist, they tell you this is actually counterintuitive. It cannot be because naive T cells are part of initiating the immune response. CTLA-4 is a marker of regulation. It suppresses the immune response. So that cannot live in the same cell. Okay? And another reason why that was interesting or weird, depending on how you think about it, is that it was actually very, very lowly abundant. So on average, only 30 cells out of roughly 1,500 cells per sample on average actually express this CTLA-4 cell. Now, just to show you how low variance or how subtle this signal is in the data, here you see a state-of-the-art representation, standard representation of the cells, and you just don't see any of those clusters. Okay, so that approach can definitely find some subtle variation in data, but is it real, right? That's a big question. So we took that result, that model, and first we applied it to other PBMC data sets. a bunch of them, and they all showed that same cluster in naive T cells. We applied it to other lymphatic tissues in human. We still see that cluster. At that point, we ask ourselves, okay, we learned that in the context of age. So we actually expect this increased heterogeneity with age to correspond also to changes in the composition of this cluster, right? And we see exactly that. So when we take either the Perez et al. Is the trained data, and Yazar et al. Is one of the test data sets used, you can see that the two box plots, the blue and the orange ones, are different in their CTLA-4 abundance when we look at the fraction within these naive T cells. So it's actually increasing in abundance with age. Next, we took it to some mouse data, and we asked, do we see those clusters in mice data? And in collaboration with Neuroself Lab in Weizmann, we actually had access to such data where we could see the clusters forming also in mice. And interestingly, the abundance of those clusters increasing with age. And we also have a few follow-up experiments we already ran in the lab, confirming that those cells, based on cell sorting, actually do exist. Actually, we do have naive T cells that express CTLA-4, and they actually increase in composition with age. And that we produced in the lab too. Okay, so a takeaway about this single cell work. So first, I showed you a model that I think of as a condition-centric cell representation. The idea is, don't let the model dictate what is in your representation. Don't just let the dominant variation necessarily dictate what you see, but rather you want it to be centric around the condition that you study. That's the idea. And that really allows us to discover this lowly abundant CTLA-4 expressing naive T cells. Now I showed you this group of clusters here, and I only talked about one, but actually we had 10 other clusters that replicated in other data sets and also increased in abundance with age. And when you think about it, we can look at obviously other clusters, but also other cell types, other tissues and conditions. So I think there are a lot of opportunities here. And also, what I showed you, this contrastive learning approach is actually part of a broader general framework for how we analyze single cell data more robustly. And I didn't show you, but we can actually apply it, and we did so for other applications, such as differential expression, differential composition. And interestingly, what we show is that you can actually take something as simple as a t-test and do drastically better than the most commonly used methods for differential expression, differential composition. So robustness is actually way more important in some cases than model sophistication. Okay, shifting gears, moving now to from cells to patients, right? This work I'm going to show you based on collaboration with folks from UCSF. Specifically, we had access to 2 pediatric asthma cohorts and whole blood DNA methylation. And asthma is actually very interesting because there are a few asthma endotypes. There are subtypes that are characterized and are the known causal mechanism of the asthma condition. So a question that we wanted to ask is, maybe those endotypes are actually more heterogeneous than we might think. And the idea is conceptually going to be very similar. Now we're not going to have cells. We're only going to have those samples by methylation sites because it's not single cell resolution. It's going to be tissue level bulk. But the idea is still the same. We want to learn some low dimensional representation, this latent representation, based on which this will essentially be our contrastive representation, based on which we can try to probe something about the heterogeneity among cases. Now, in the single cell example, we had this max-min formulation, and the minimization was across subsamples of the data. Now, the reason why we could do that for single cell is because we could actually take small subsets of samples, hope that there will be some of them balanced with respect to unknown confounders. And at the same time, we were powered enough because we had many cells per sample. That's not the case for this DNA methylation example, right? So we cannot take small subsets of samples because we will not be powered enough. We don't have many cells. We just have one data point per sample. At the same time, we don't want to take large subsamples of patients because there's no chance they're going to be balanced, right? So we do need to take a different approach. And the idea here is actually to go back to what I told you earlier about unsupervised learning, capturing dominant source of variation in the data. So this is what, for example, as an illustration we might get from the cases. What we can do is we can actually apply a similar model to data from controls. And now we can actually look for consistency in the patterns that are forming. And if we're willing to assume that disease heterogeneity is actually a variation that is specific only to disease cases and not to controls, what we can do is we can look for the shared variation between these two representations, meaning shared sources of variation between cases and controls, and completely remove those from the data. Remove them from the data of cases and controls, and we can still try to maximize this objective of ratio of variances. We call that phenotypic aware component analysis, or PACA for short, and we apply that method to this DNA methylation data. Specifically, we had these two cores, one from Latino, it's a Latino pediatric core, the second one is African American pediatric core. The way it looks like is we have the cases and controls, we apply the model based on this discovery data set. And then because we learned just one component, it's just one-dimensional latent representation, we could actually look at the scores of each one of the patients, and we also bin them into different quantile groups. And then we took the replication data, we applied the same model. We also had those scores, one value essentially per patient, which we then bin to those quantile scores. Okay, first question, what is this quantile score really capturing, right? So here, what you see in this red plot is a few biomarkers that are interesting in the context of asthma. Specifically, there are two blood biomarkers, blood eosinophil counts, IgE, there are 4 metrics of lung function, and there is even the BDR, which is response to bronchodilator. Essentially, it's a metric of drug response. What you see here is essentially the mean levels of those variables when you just look at patients with very low DNA methylation score based on the model. Those are percentiles 1 to 20. And what I'm going to show you now across the x-axis is what happens, how the joint distribution of those clinical variables are going to change as we increase the score of patients. And you can see that there is a very clear shift in this clinical presentation as we go up in these percentile groups. Interestingly, that's replicated really, really nicely in the replication data. So we see the same shifts. Now, at that point, we also noticed that two big factors that are changing are actually the body eosinophil count, the PCE and the IgE. And the reason why it's interesting is because there is actually an asthma endotype, type 2 asthma endotype, for which those are the most clear biomarkers, right? So are we simply recapitulating something that is known, an endotype that is known? That was essentially the next question, right? And the way we tackle that is by saying, okay, let's look at non-type 2 patients separately from the type 2 patients, right? And see if we find variation within each one of these two groups. When we look at non-type 2 patients, and now we're slightly changing the clinical variables to those that are even more interesting within type 2 asthma, we actually visually see that there is no big difference between patients that have below median DNA methylation score or above median DNA methylation score. But when we look into patients that are type 2 patients, we do see pretty dramatic clinical presentation difference between these two groups. Okay, so now we can really focus on these two type 2 patients and try to ask, what is the meaning of this DNA methylation score within that specific subset of patients, right? And because we have DNA methylation data, we could ask that question from the perspective of DNA methylation, and specifically it was bulk data. So we applied our deconvolution method, and we actually found that there are quite a few changes that are specific to eosinophil. So eosinophil specific changes in methylation that happen in those patients that have high DNA methylation scores. Those results replicated, and we also collaborated with Catherine LaPrize, who actually collected DNA methylation from purified eosinophils from asthma patients, and we could actually validate. Next question we asked is, it's actually multi-omic data sets that we had. So we had gene expression. Can we look at changes in this gene expression? What we found is that many of the genes that actually change with this DNA methylation axis within these type 2 patients were actually genes that are known to be involved in the type 2 inflammatory pathway, which is interesting because when you think about it, those genes are actually also separating between the non-type 2 and the type 2 patients, right? So the next question was, okay, what are the genes that are actually not differentiating between type 2 and non-type 2, and at the same time, differentiate between high and low DNA methylation score patients, right? So now we're looking for a signal that is really specific to type 2 patients. It does not separate them from the non-type 2 patients. It's only variation that is specific to type 2 patients. And what we see is that there is this B cell specific module of genes that are highly expressed in that specific top right quadrant. Those are genes that are known to be involved in activation, proliferation of B cells. We also see change, increase in B cell composition, and those results also replicated. Now, the reason why it's interesting is because state-of-the-art drugs today for asthma patients are actually drugs targeting the type 2 inflammatory pathway. And those drugs are very costly. They cost in the order of $40,000 per year per patient. And you're only eligible to get it if you hit two thresholds, roughly speaking, two thresholds of two biomarkers, such as the ones that we use to separate the type 2 and non-type 2 patients here, right? So next we ask, is it possible that this stratification is actually informative of drug response? So we took data from randomized clinical trial that also had gene expression levels measured from the samples. And we actually found for this one particular drug that for which we had data, omalizumab, that I-score patients, so I-score expression in that case, because we didn't have methylation for those samples, are actually the ones that poorly respond to omalizumab. Interestingly, more than that, not only that they poorly responded, they actually had even higher asthma exacerbation, which was the outcome of the trial. Okay, so I'll take a couple of takeaways here. So I showed you how robust contrastive representation can reveal this unknown subgroup within type 2 asthma patients. And I showed you that there is this eosinophilic specific epigenetic reprogramming among those patients that have the I-scores that comes with some elevation in B-cell activation proliferation. And there is, and that subgroup seems to not respond to this particular drug, and we suspect that that might be the case for other biologics as well. It seems like those two biomarkers, the BEC and the IgE, are not really great biomarkers for responding to those anti-type 2 inflammatory therapies. And when we think about it, we really should try to revisit those eligibility criteria because of that, and ideally try to integrate and combine those blood biomarkers with molecular information. Okay, so I showed you a couple of examples where this idea of patient stratification works really nicely. There are some other examples that we also found. But at the same time, you know, if we go back to this question, can we do that systematically, right? The answer is still no, because when we apply this methodology to some conditions, some data types, it doesn't work. And just to give you one example, we took data from all of us. So this is EHR data, roughly 70 features, demographics, prescriptions, ICD codes and so on. And we asked which glaucoma patients will need in the future surgery because of their glaucoma condition, right? And what you see here is taking the contrastive representation learned from 4 different models. The two ones on the right are the ones I presented to you. And you see area under the precision recall curve. Now, it seems to be working fine, and that's based on held out states. But when I take states that were not part of this evaluation, I see that, again, performance drastically drops, right? More than that, if we now evaluate same metric, area under the precision recall curve, we evaluate that under different demographic groups. What you can see if you focus on each one of those columns separately, that there is a lot of instability between different demographic groups. So the model tends to perform very differently for different demographic groups. So that's another way to say the model does not generalize very well across populations, across location, and so on. So the method didn't really work well enough in that case. So the big challenge is really how do we take this idea that appear to be working really nicely on those few examples, find some exciting findings, and make it into something that is even more robust, can more systematically work for this vision of patient stratification. And the idea of my future work is exactly to do that. I'm going to use the foundations of an active R21 brand that I've been the PI on for really learning and developing those foundations of those contrastive learning models for patient stratification. My first research program will really focus on trying to make that more broadly applicable. And specifically, that will allow us to answer questions such as which patients will benefit from an intervention, or maybe we can identify some molecular variation in those patients that is specific to them. And what the first thing to do, again, is making sure that those robust contrastive learning models can really be applied to multiple conditions, right? And to multiple data types. And I showed you this example with glaucoma. Now, the one thing I didn't tell you is the method to the right, the RCL, which is the one I showed you in the context of single cell. Originally, we used subsets of samples. That's what I told you, right? For the EHR data, here we had in the order of 5,000 patients. We cannot subsample patients for the same rationale I told you about the DNA methylation. What we did do instead is to ask the model to be robust across different states, so patients coming from different states. That didn't work. So that led us to the insight that there are actually systematic confounders even across states. So instead of trying to be robust across states, we need to find data domains, that is just one of them, or data splits, if you will, across which we want our model to be robust. And that's exactly the second aim of this research program. How do we define those domains, which can be a combination of how drugs are being administered. Maybe there is a temporal component, maybe that changes with time. Where the clinicians were trained, right? There could be combinations of unknown factors that will essentially be confounded. How do we learn those domains that will capture those confounders in an unsupervised way, because we don't know them, and make sure the models are robust to those domains? And we already had some nice, interesting success by doing so. And what you see here is exactly that. We were actually able to define in an unsupervised way domains for this glaucoma application, where eventually we were able to almost completely balance the training test performance. And if you look at the rightmost column in the table, you see that the performance across demographic groups becomes much, much more stable now. Now, obviously, we don't want to do that in isolation. We want to try to tie that to an interesting clinical translational application. At that point, we contacted Sally Baxter from UCSD. She's an ophthalmologist. She told us, you know what? The one thing I don't know about a patient who I just diagnosed with glaucoma is what should be the first-line treatment. I'm just guessing. So we asked, is it possible that the patient certification score we learned is actually indicative of what a patient should be getting? And what we found is that high-score patients, particularly those with the high quartile score, are the ones that are more likely to benefit specifically from prostaglandins as a first-line treatment in terms of how likely or less likely they become in the future to get surgery due to glaucoma. And another component of this research program is really about the point of molecular variation. And specifically, I showed you this example. To this part DNA methylation data, which is two-dimensional, it's tissue level, and we infer this information on the cell type hidden axis of variation. And by now, there are quite a few deconvolution methods that can do that. They can take the cell type level hidden variation and associate that with a disease or a condition. When you think about it, this is just a static type of statistical association. And in clinical practice, clinicians actually find clinical biomarkers in many cases more useful when they're measured for the same patient over time. So if you think about something like GFR, PSA, CBC, all those biomarkers, they tend to have more information when you look at their changes over time rather than just at the absolute levels. And I think that's a missed opportunity right now in genomics. And we're starting to have data sets that do have, for example, multiple time points that we can start exploring that idea. And specifically what I want to do is to develop other types of deconvolution models that can take those rich data sets, those large clinical genomic data sets that have thousands of bulk level samples, and try to infer at the cell type level information that takes into account temporal variation. One example for that is time-to-event modeling, time-to-event deconvolution models. Another example is time series deconvolution. And we can also try to incorporate genetic effects. One example for that, we actually did implement this preliminary model of time-to-event deconvolution. We had DNA methylation from blood drawn from patients at UCLA during hospitalization, and then they were followed up after discharge. And we asked who is going to develop sepsis and when. And we actually were able to find a CP gene, this particular gene, MYO1E, that had a very significant and very dramatic predictive power of post-hospitalization sepsis. And specifically, we were able to attribute that statistical signal to two specific cell types. Now, the first research program actually has a few limitations that are pretty important when you think about clinical practice. And that's exactly what I want to address in the second research program. Specifically, if you think about the glaucoma example, we have our patient certification score, and I already told you that in our model, eye patient scores are more likely to respond to prostaglandins as first-line treatment compared to other patients. But when a patient goes to the ophthalmologist doesn't only want to say, okay, that patient is more likely to benefit from prostaglandins over the other patients, so let's give them that. What about other drugs? Maybe that patient is also, is even more likely to respond to, say, beta blockers, another first-line treatment for glaucoma. So can we direct our models to ask more specific questions? Now, if that sounds familiar, you're not wrong. We have the supervised models to do that for us. And you can think about this red dot here as the optimal model for asking whether the patient is going to respond to beta blocker. Now, what's the problem with supervised models? I showed you at the very beginning a few examples, and there are many others where models don't just tend to not generalize, right? And when I think about fitting those contrasting models, I actually think of it from that perspective of supervision as a way to cut the search space. Because when you think about whether the patient will respond to a specific drug versus the other, this is naturally a contrastive type of problem. There must be some variation that is more enriched among cases, right? So when you think about it, if you look at the intersection between supervision and contrastiveness, then the optimal model is likely to kind of live in that space. So that's the idea. And clearly we have to find the right balance between the level of contrastiveness and the level of supervision. We also have a couple of other trade-offs that we have to balance. First is robustness versus sensitivity. So the max mean is actually very rigid when you think about it, right? Maybe we can change the mean to some quantile function, for example, so we're not maybe overly trying to be robust. And second is generalizability is typically great, but it's not the right thing to do always. Maybe in some cases there is a population that has some disease subtype that is specific to that population. So we might miss that if we try to generalize across everyone. So we also have to figure out how to balance between generalizability and specificity that is actually real and replicable. Okay, finally, I want to say that it's really important for me to develop those programs to be highly collaborative. The idea is to really try to make an impact, work on problems that really matter, that can make an impact on patient lives. It has to be collaborative, obviously. And when I think about innovation in biomedical informatics, I really think about this pipeline. So we start with translational discovery. That could be, for example, prognostic biomarker. We then move on to a step of validation, optimization. Maybe you want to run a clinical trial or prospective trial to make sure that it's real. And then eventually we have to find a way to effectively, efficiently integrate that into a clinical decision-making process to make sure that can actually be delivered to patients. And just to frame everything I've shown you in the last hour, I think of my work as really focusing on the link between these two components. Because of this big problem of generalizability, we really need to improve that link to make sure that more of our discoveries are really, they really keep moving on this pipeline. And I think in the context of Beamer, I think that's the perfect place to do that because there is all this expertise along this pipeline and for this entire pipeline to actually get it done. And with that, a few acknowledgement of a few people, key people in those work that I didn't mention so far. First one in the single cell work, Samuel Berry is actually sitting here from Stanford. Yixing Wang from Michigan, they helped with the analysis and the work on the single cell application. Shlomit Reichzeriger from Weizmann did the experimental validation with the CTLA forces. The asthma work was led by Aditya Gorla and Jonathan Mitonsky. And the glaucoma example was led by Arush Ramtek from UCLA. And thank you all for coming and for listening.
Questions from room before I take the online questions? Yes, let me begin with my question and then we'll move on to others. So you talked about like tractability and generalizability, right? And to what extent you've seen those goals conflict and how you manage that conflict?
I'm not sure they conflict. I mean, I see them as two different concepts that I just try to combine. I mean, let's see. I guess in the context of tractability, seeing as, and that's just one way to see it as, let's look for the subtype that is more likely to show signal, then I think, generalizability becomes a factor because, you might find a patient subgroup for which you can learn much more accurate model compared to what you can do for the entire population, but then it will not generalize. So in that sense, these two are connected. So you really want to try to combine them and not just go for one of them in isolation. Of course, generalizability is important for everything, not just for patient stratification, for any type of modeling. We should obviously strive for our models to be replicable. Yeah.
Questions, so we have, okay.
Yeah, that was a really nice talk. So I'm curious, you talk a lot about generalizability, tractability from the perspective of like predicting from a single modality, let's say DNA methylation or transcription or maybe an EHR. What are your thoughts on building multimodal models? Is it just gonna be additive in performance, or do you think there'll be synergies, and what are the opportunities do you see there?
Yeah, great question. And definitely that's one of the kind of the to-dos. And it's really a question of what additional signal you get in one modality versus the other. And there are some technical challenges, obviously. First of all, because different data sets are different, but also there is also a general challenge with just building richer models, and multimodality, automatically means richer models. And the reason is, if you think about kind of more standard representation learning methods, you can think about an autoencoder as an example for a popular architecture. So you encode your input, and then you decode your input back to the original visual space. The reason why you can learn those really rich models and over-parameterize and still think will work, because we have very, very strong globalization by the fact that you try to minimize the error between the reconstructed and the original input. Now, if you are looking for contrastive variation, which is really subtle variation that is buried somewhere, in the data, you cannot use that trick. You cannot regularize by looking for minimizing reconstruction loss, because reconstruction loss, by definition, will be minimized by leveraging dominant source of variation. So we have to be very creative, and yeah, I have a couple of ideas how to.
Hi, I'm Besat from Mussok Lab. I'm a biomedical data scientist. Very impressive and interesting work. My question is about, you talked about finding the variations between the positive case and the control using, for example, subsampling learning and contrastive learning. I think this is, when you are working with a limited number of patients, and again, we don't have access to the whole population again, I think this is more related, my question is that how this difference between the variations lead to generalizability, because I think this is more related to uncertainty of the model, and maybe the subsampling and contrastive learning will have a great impact on reducing the uncertainty, specifically the epistemic uncertainty of the model. And I'm wondering how this will lead to increase the generalizability of the model with a limited number of the patients.
Yeah, so the way I think about this contrastive approach, the contrastive representation, we may be interested in other type of representation, not necessarily contrastive. Contrastive is a way that I think is very powerful for specifically the task of patient stratification. And then we bring this generalizability aspect, but I think we should bring it to any type of application. But that being said, we might be interested in other types of models for which contrastive learning will not make sense. So it's not like, let's kind of marry to that idea and apply that to any idea. The idea was not to use contrastive representation for replicability. The idea was for the patient stratification. And that the subsampling, as an example, was a way to induce this generalizability so that the stratification we find will actually replicate. If that makes sense.
Thank you for the excellent talk. I actually had some questions, like the majority of the clustering algorithms you applied are to genomics data, which are pretty homogeneous and work quite well. But most of the times in our data sets, we don't have any genomic data sets. I was wondering if you had any guidance, any advice for doing clustering our direct EHR data, because they rather underperform. If you have any experience dealing with clustering those data.
Yeah. So, the common example is one of them, right? And when you just run k-mean clustering of PCA, and that's typically the same for the more, modern deep learning based representation learning method. Again, unless you try to do something differently, you will capture dominant source of variation. In EHR, that can be demographics, level of engagement of the patient with the health system, and so on, right? So, you really need to I think, talk about a specific task you want to like, what is it that you want to do, and try to kind of fine tune the algorithm to what you want to achieve. Because, in many ways, just clustering using, a standard unsupervised learning method, sure, sometimes it will work, but, you don't have any reason to think why it will systematically work. Because it's not like you're systematically targeting particular signal in the data. You're basically telling the model, please tell me what's the dominant variation in the data. And sometimes it might be clinically or biologically useful, but generally speaking, it's not going to be, right?
That's exactly, I mean, I wanted to know how to refine based on this outcome. Like, imagine we want to predict mortality in a group of patients with only EHR data. How can we be sure that this pattern that is being predicted is actually relevant to that and not a
Oh, you're talking about evaluation, or how do we actually learn the cluster?
evaluation? Because as you know, when you are doing the clustering for the genomics data, you can be somewhat more certain of the results because it's a homogeneous data. It's the clustering actually creates pattern. But let me put it this way. Like when the clustering algorithm separates patients based on age and sex, the certainty you gain is not the same as like the cluster of genomics that are expressed together.
Yeah.
I'm wondering how to connect that bridge more with when we just have the EHR.
Okay, so if I got you right, and you're talking specifically about evaluation, so I think we should be very careful. And let's take the common example which I've shown. What we did was to take patients coming from some specific geographic location of the United States, and they were completely held out. And once we learn a model, we ask, okay, what's the evaluation even between states in that left out geographical location? And also within that geographic location, let's look at how it performs across different demographic groups. So you really want to kind of try to break it down and see, how it performs, right? So that's kind of purely in terms of performance. If you're asking about the formation of clusters, that's a different story. But taking that approach that I've shown, you could do maybe something like, let's take those patients who had, were on the kind of the bad side of mortality, and those who were on the good side, and then try to treat them as case and controls. Try to learn some patterns of heterogeneity that are specific to those who have, earlier mortality, just as one example.
Thank you so much.
So when you run this framework and you identify these different strata, I'm wondering if there's a need to identify which of these are clinically significant. I know that you had shown like you do some analysis and like this was something unexpected, for example, right? I think some certain gene expression that was unexpected. And so I think there was some deep dive into that, but I'm wondering if there's a way of like, if there's even a need to do that. Does that make sense? Because like, I'm wondering just like how sensitive the algorithm is to all this kind of variation, and if there's like 100 different phenotypes that emerge, is it really scalable to like process all of that?
So, okay, so you're asking how to essentially take all the clinical phenotypes we have and figure out what is the right one, what is it that we're capturing, or
because I'm just wondering if like some of them aren't really clinically relevant, for example,
Sure,
and
yes.
then like clinicians end up looking at that, right? And it's like you don't want to waste their time, of course. So that's why I'm just trying to understand like, what the workflow is.
Yeah, so I can give you the specific example with asthma. We actually started by looking at a bunch of clinical phenotypes. And those red plots I showed you, it's those are some of them, and just ask, okay, what's different, right? One of them was this BDR, the bronchodilator response. This is a response to albuterol, that's basically the inhaler that asthma patients sometimes need. And we found that statistical association, and it actually seems like those other biomarkers are predicting this BDR only for high school patients. So we were really excited about that result. If we got to the clinicians, they told us, okay, that's actually very nice, that's very cool, but it's not clinically meaningful because that's a cheap drug. It's always going to be first line drug, doesn't have any adverse effect. Let's do something different. And then we converge to looking into this type 2 inflammatory outcome. So I think a lot of times, from my perspective, it's really working in close collaboration with the experts, with the clinicians to make sure that we are targeting the clinically meaningful questions. You are right if you also imply that the models may not necessarily target the clinically meaningful question. And that is one of the limitations. That's exactly what I present in the second research program. How do we try to direct the models to tell us more of what we want and less give them the flexibility to decide which contrastive variation to capture?
Okay, so we don't have time for any more questions, but thank you, Elior, for your talk, and if you have questions, feel free to just hang around to ask more questions. I think we have time. Sure.